And there's the ding-a-ling. Okay. I've been putting all of this in the framework of animal classification as we currently understand it. And sorry, one moment, there's somebody I need to let in. Not all groups of animals, of course, have parasitic members, and many others have only a few. So, of course, there aren't any parasitic sponges that I'm aware of. Most cnidarians are not parasitic, but there are the myxozoans. We can divide animals that are more complex than that into protostomes and deuterostomes. There aren't many parasitic deuterostomes except for parasitic vertebrates like lampreys and vampire finches and those awful eels that live in shark hearts. In protostomes, the lophophorates don't have any known parasitic members. Trochozoans don't have many except for the leeches. Platyzoa, we've talked about, that's flukes and tapeworms and monogeneans and aspidobothrians and acanthocephalans and maybe the mesozoans. And then ecdysozoa, ecdysis is the ancient Greek word for molting, shedding your skin. The journalist H.L. Mencken coined the word ecdysiast to be a more lofty synonym for a stripper. But in biology, ecdysozoans includes animals that have a cuticle that's stiffened with chitin. Chitin, the stuff that makes cockroaches go crunch when you bite them, and that they periodically shed. And there's a number of ecdysozoan phyla. This includes the two largest, the arthropods, with something on the order of something between 750,000 and 1 million known members. Now, it's probably more than that now that I think about it. Well over 1 million known members, and the nematodes, probably the second most diverse phylum that there is, together with a group called horsehair worms that you can see in Arkansas sometimes, and some others that aren't parasites, so we don't really have time for those. So what makes ecdysozoans are a cuticle with chitin. The chitin is molted, and usually it's molted a fixed number of times. It's not molted indefinitely throughout life. Insects like cockroaches, for example, will hatch into a juvenile and then molt maybe, I can't remember the exact number, but I think it's 4 or 5 times before reaching their adult size and morphology. Roundworms typically molt exactly 4 times in their life cycle, and molting is under the control of hormones called ecdysteroids. I should mention that life cycle stages between molts are known as instars. So we can talk about a first instar juvenile of a cockroach or a second instar juvenile or whatever we're talking about. They pretty much ditch cilia. There's never any cilia on the outside of the body. They never swim using cilia, and in most cases, there aren't any cilia present at all. Roundworms, there's like one that I know of that might have a few rudiments of them, but the vast majority of roundworms never develop any cilia at all. They don't even have flagellated sperm. The sperm can't swim. The sperm look like little amoebas. And because the sperm can't swim, arthropods and nematodes can't reproduce the way that a lot of marine invertebrates do. They can't simply fire lots of eggs and sperm into the water and just kind of let nature take its course. Arthropods and nematodes usually have to copulate to get the sperm to where they're supposed to go, because the sperm can't swim on their own. So there's often lots of fairly complicated genital apparati needed to get the sperm close enough to the egg for them to crawl the rest of the way. And then anyway, the group was established in 1997 based on shared similarities in DNA sequences. And as I mentioned, there are click, 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 maybe 6 or maybe more like 8 or 9 ecdysozoan phyla. The 2 main parasitic phyla are the Arthropoda and the Nematoda. We'll talk a little bit about a third, the Nematomorpha, aka the horsehair worms. Other than that, there's really not very many parasites in the rest of the group. But arthropods and nematodes are 2 of the most important parasitic taxa by far. And you're familiar with the leftovers of the ecdysozoan life cycle. That's the molted exoskeleton of a New Zealand cicada. Of course, we get molted exoskeletons of our own cicadas in summer, which, you can pick off the trees and like make perch on your fingers or perch on your nose and, run around freaking out your little sister. Not that I ever did that. But yeah, you're familiar with this process of ecdysis. So from there, let's jump right into the Nematoda. There's about 28,000 described species. That's not the largest in terms of known species, but the true number of species is estimated to be many times that. In some ways, they're not that easy to study because telling species apart can rely on some very subtle features. And again, the total diversity is probably much, much greater than 28,000. You can find free-living nematodes everywhere you can think of. There are nematodes in the oceans. There are nematodes in soils. In fact, they're the most abundant animals in soils. There are nematodes crawling through the soil outside my office window even as we speak. If you take invertebrate zoology with me, we go find some. There's nematodes in pond mud. There are nematodes in decaying leaves and compost piles and mosses and things like that. It's been estimated that 4 out of 5 individual animals on Earth are in fact nematodes. That's how abundant they are. And there are lots of free-living ones. Parasitic ones infest almost everything you can think of. We won't talk about plant parasites much in this class, but they cause 100 billion annually in agricultural losses. Every crop can be subject to nematode attack. This is why, by the way, some of our larger agricultural universities, University of Nebraska is one that I'm familiar with, actually offer PhDs in nematology. And Arkansas actually has an office of the state nematologist. They're located down in Hope. And that sounds weird. Why would the state spend perfectly good money on having an official state nematode expert? But when you realize that that's mostly looking for plant parasitic nematodes, you see why it's important. Farmers can send soil samples to the state nematologist, and the state nematologist can recommend control measures if necessary. So we won't talk much more about that, but that's a really big deal. There are animal parasites that are parasites of just about every other animal that there is, including every economically and socially important animal. You veterinarians in the making will encounter these. There's nematodes that infect horses, dogs, cats, goats, sheep, all of the domestic critters. And of course, they infect wild animals as well. And then human parasites, pinworm is probably the most common parasitic disease in the West. Hookworm is a big one. Ascaris is the human intestinal roundworm that can block your intestine if the infection is gets too severe. Trichinosis, elephantiasis, river blindness, guinea worm. Some of those are tropical diseases that you may not encounter, but there are several that you could get here. I always like starting with Nathan S. Cobb, great nematologist of 120 years ago, who complained that the nematodes, poor beasts, do not furnish hides, horns, tallow, or wool. They are not fit for food, nor do they produce anything fit for food. Neither do they sing or amuse us in any way, nor are they ornamental. In fact, when they are displayed in museums, the public votes them hideous. However, he's the person who realized just how abundant nematodes are, and he concluded that if you could get rid of all of the matter in the universe except the nematodes, our world would still be dimly recognizable. And if, as disembodied spirits, we could then investigate it, we should found its mountains, hills, vales, rivers, lakes, and oceans represented by a film of nematodes. The location of towns would be decipherable, since for every massing of human beings, there would be a corresponding massing of certain nematodes. Trees would still stand in ghostly rows, representing our streets and highways. The location of the various plants and animals would still be decipherable, and had we sufficient knowledge, in many cases, even their species could be determined by an examination of their erstwhile nematode parasites. That's how many nematodes there are. They're everywhere. They're typically small, ranging from microscopic in size to a few millimeters in length. Some of the parasites get pretty big. The human intestinal roundworm, typical ones there could get you maybe, I'm thinking about 4 or 5 inches, maybe 10 centimeters or so. Those can get pretty big. A parasite of sperm whale placentas, because of course there has to be something in sperm whale placentas, right? If you have worms in hippopotamus eye sockets, you have to have something in sperm whale placentas or the world wouldn't make any sense. Placentanema gigantissima can reach 25 feet. That's a pretty big nematode. There isn't much of a visible, typically there's not that much of a visible head. Both ends of your typical nematode tend to be pointed. There can be some head structures, but it's not usually a big swollen head or anything like that. There's not that many obvious sense organs. The body is filled with fluid. There's a spacious body cavity called a coelom for technical reasons that I'm not going to go into. It's sometimes called a pseudocoel because it's not lined with tissue derived from a group of cells called the mesoderm. I'm not going to say much about that. The body's got bands of longitudinal muscles, but no circular muscles. What that means is nematodes can flex side to side, but they can't make any part of their body narrower. Earthworms and a lot of burrowing worms can use that big fluid-filled body cavity and have their muscles work against it because fluid is incompressible. So they can make parts of the body wide and parts narrow and have these peristaltic waves of wide and narrow passing down the body and burrow that way. Fluid used in that fashion is called a hydrostat. Nematodes can't do that. Nematodes can't inch. They can't make any part of their body more narrow or more wide. All they can do is thrash back and forth. Makes for a very distinctive kind of sine wave bending of the body from side to side, kind of like Axl Rose in the video for Sweet Child of Mine. You may not have seen that, but there it is. So the method of locomotion is very distinctive. There aren't any functional cilia in or out of the body. The mouth is typically triradiate, meaning it looks kind of like a three-pointed star, like a letter Y or a Mercedes logo. The mouth leads into a muscular tube called the pharynx, Often at the end of the pharynx, there's a bulb. So the pharynx acts kind of like an animated pipette or eyedropper, because nematodes typically feed by suction. They can't open the mouth very wide. They generally can't, well, there's some that have jaws, now that I think of it, but they can't really bite off chunks. What they'll typically do is suck up food, which might be bacteria if it's a free-living nematode. It might be plant juices if it's a plant parasite. It might be, blood or other bodily fluids if it's an animal parasite. There's a nervous system. There's a ring of nerves around the esophagus that gives rise to 4 main nerve cords. And there's also a tail extending past the anus. And thus you may know the nematodes. And this is probably the best known. And if any of you have ever hung out much with Dr. Ferris, you've probably heard of it. This is Caenorhabditis elegans. It's a free-living nematode. It lives in soils. It tends to like more organic, rich soils than your average dirt. I don't think I've ever seen it in typical soil samples, but it can be very abundant in compost piles, for example. And it's abundant in biology labs because it's used as a model organism in cell and developmental biology. Dr. Ferris uses these in her studies of aging. And you'll encounter this a lot if you go on to research, especially in developmental biology. But I'm showing it here because you've got the basic nematode features. The pointy end at the top is actually the tail. The head is down at the lower right. Within the body, you can see a fairly thick-walled tube. That's the pharynx. And you can see there's actually a couple of bulbs if you trace the gut back. Much of the grainy stuff that fills the body is ovary. On the inner side of that largest bend, there's actually an embryo inside an egg next to the female genital aperture. It's not at the tail end of the body, it's more or less in the middle. And the anus is kind of on the outside of that last bend, and that whippy, pointy part that you're seeing is the tail. So if we diagram this, the empty space inside the gut is in orange. The gut is in dark blue, and you can see it's got those 2 bulbs leading into a stomach. There's an excretory cell called a flame bulb in kind of that light blue or teal, and that nerve ring is in dark green. C. elegans, that particular one, is one of the few animals where the sexes are males and hermaphrodites. That's called being androdioecious, and it's not very common in the animal kingdom. Up at the top, you see a male, and the male's got that big blue testis filling up a good chunk of his body, and at his tail, instead of a long whip-like tail, he's got a pair of flaps making up what's called a copulatory bursa. Those flaps will actually wrap around the female's opening, and sticking out from between the flaps, you have copulatory spicules. Those will hold the female's genital aperture open so that he can transfer sperm. Hermaphrodites will start by making sperm as juveniles, and then they'll switch over to egg production as they mature. They can self-fertilize, but they will mate with a male if they get the chance to. They don't often get the chance to. In the wild, hermaphrodites outnumber males by 20 to 1. But in the hermaphrodite down below, the female organs are active. That's an ovary in pink, and you can see egg cells in various stages of development, including some embryos. Close to where that uterus opens up, you can see some embryos that look to be at about the 8-cell stage. There's a male with its copulatory bursa. That's light microscopy on the left, and then on the right, that's a scanning electron micrograph. You can see the two fans that make up the bursa, and then sticking out, you can see the copulatory spicules that in the wild would be inserted into the female's vulva to guide the amoeboid sperm in close to target because they can't swim. And again, males make up only about 0.5% of a wild C. elegans population. Hermaphrodites can self, but they'll mate with a male if they get the chance. Okay, a nematode egg will hatch into, this stage calls them larvae. Probably a better word is juveniles, because they look pretty much like miniatures of the adults. There's some differences, but they're not really vast. You know, very much unlike caterpillars transforming into butterflies. You know, caterpillars don't look anything like butterflies, but juvenile nematodes look a lot like adult nematodes. And they'll molt exactly 3 times, producing 4 instars that are numbered. I prefer J1 through 4, but you'll also see L1 through L4. So there you've got L1, L2, L3, L4, and then the adult. The instars look more or less like small adults. The J4s will molt 1 more time to generate the adult stage. And if you look at the lower right, L2 or J2 instars can actually switch over to an alternate pathway and molt into a stage called a dower. The dower is a resistant stage. Dower is German for enduring, and nematodes will switch over to a dower stage or a dower larva if conditions are harsh. The mouth is often sealed shut. The intestine will shrink. The muscle may shrink. Arrested germline development means that there's no more development of ovaries or testes. Dowers often are very still, although they can still move if you stimulate them, if you heat the slide or something like that. In parasitic nematodes, the dower is often the infective stage. There are species of soil nematode where the dower larva will stick its tail to a blade of grass or a blade of moss and just sit there sticking straight up, waiting for a host insect to come by, and then the dower will jump onto the host insect. The dower is often the infective stage when you're looking at parasitic nematodes. Okay, there's a cuticle that is periodically shed, of course. There's a surface coat of glycoproteins and carbohydrates, and in parasites, that may be important for counteracting the immune system of the host. Below that, there's a lipid-rich layer called the epicuticle. Below that, the cortical layer is rich in a cross-linked protein called cuticulin, as well as chitin. You'll see there's a feature there called the annulus between 2 annular furrows. Nematodes aren't truly segmented, but they often have ring-shaped ridges on the cuticle, and I'll show you what that looks like in a bit. Below that, fibers of the protein collagen, right, the same kind of fiber that makes up your tendons, wrap around the worm, and they wrap at 75 and 135 degree angles to the long axis. That's a very strong arrangement. The worm can bend without forming kinks. I do believe I've seen this kind of arrangement in things like garden hoses. If you get a garden hose that's made up of fibers that are wrapped at these diagonal arrangements, it won't kink up. And when you turn on the tap, the water will always come out the hose. You can't get these kinked up bends in it. That arrangement can handle high pressure in the worm's body cavity. And the worm can be very flexible, even though the fibers are relatively stiff. If anybody's ever had one of those finger traps, um, it's like a tube made up of woven, I think, bamboo or grass or something like that. You put your fingers in it and you pull, and your fingers get stuck. If anybody's ever played with one of those, it's a little bit like that. You can take fibers that aren't that are stiff and make a structure that can stretch, and make a structure that's very hard to break if you arrange those fibers in this kind of two different diagonal angles to the long axis. It's a very strong yet flexible arrangement. There's a typical nematode head. You can see the mouth. That black area is triradiate. It's got three equally spaced rays coming out of the center. Again, kind of like a Mercedes-Benz logo. There's lips surrounding the mouth. There's sensory setae surrounding the mouth. There's also some sense organs in pits surrounding the mouth. Details of exactly what surrounds the mouth are often important for identifying species. Within the mouth, there may or may not be a piercing structure called a stylet, like a little dagger. This is very common in plant parasitic nematodes. This one is actually a plant parasite, and you can see the stylet is actually extended out of the mouth. The stylet can be extended or can be pulled back in. There are some animal parasites that also have this. So do some free-living nematodes that are predatory on other invertebrates, like other nematodes. They may also have a stylet, but it's most common in plant parasites. We see nematodes like this in the nature reserve when we sample them in my invertebrate zoology class. Free-living ones will often have various thickenings on the cuticle. This is another one that we sometimes see in the UCA Nature Reserve. This is under your feet when you're walking through it. It's not a soil parasite. That's a mistake of mine. This is a free-living nematode. You should be able to see what I mean by those annuli. This ring-shaped ornamentation. You can see that on both the light micrograph to the left and the electron micrograph to the right. It doesn't correspond to actual segmentation of the organs the way it would in an earthworm. It's just this kind of, ring-shaped ridges and furrows in nematode cuticle that makes it look a little like a worm version of the Michelin Man. And this particular species happens to have these weird extensions of the cuticle on its head. I have no idea what these do. They've got this very odd, complicated shape. What function they might have, I am not sure. You usually don't see weird deedly bobs on the heads of the parasites, but there are some exceptions. This is Toxocara cati. This is one that you vets might see at some point. This is called the feline arrowhead worm. There's Toxocara, and there's a related genus called Toxoscaris. Cats can get both. I think there's a relative that infects dogs. And you can see that triradiate mouth with the three lips up at the top. And then surrounding the mouth, you have these three flanges, making the thing look like, well, an arrow. Hence you can see why it's called an arrowhead worm. I'm not entirely sure what those do, what function they might have. Might try to find out, see if anybody knows. But yeah, this is just an exception to the rule that animal parasitic nematodes tend to have much smoother bodies than some of the free-living ones that you see. Nematodes may have pit-shaped chemosense organs in the head. They're called amphids. These are two free-living species from Nevada grasslands, not Nevada, Nebraska. And in fact, I borrowed these pictures from the University of Nebraska's nematology website. I think I mentioned they've got a very good graduate nematology program. In the one on the left, Achromodora, the amphid is shaped like a little snail shell, like a little coil, shaped more like a bulb in the nematode on the right, Plectus rhizophilus. The tail may have phasmids, that's another type of chemosense organs, and the shape and location of these can be important for classification. The last free-living nematode I'll talk about is Caenorhabditis elegans, which I showed you a picture of earlier. And this is a context where you might encounter nematodes even if you don't ever study parasitology again. A gentleman named Sidney Brenner started using nematodes to examine developmental genetics. He started back in 1965, and after nearly 40 years, he shared a Nobel Prize for what he had done. The reason he did is that C. elegans adult hermaphrodites consist of exactly 959 cells. There's also 131 other cells that are produced during development, but they die by a process called apoptosis. If you don't know what apoptosis is, Dr. Runge is a specialist in apoptosis. That's what he studies in cell biology. And apoptosis is also known as programmed cell death. A cell undergoing apoptosis basically does the equivalent of turning out the lights and putting the chairs up on the table, shuts down, and breaks up into tiny little blebs and disappears. The reason you don't have webbing in between your fingers and toes is that the cells that used to be there died by apoptosis when you were still an embryo. The reason why frogs don't have tails is that their tail disappears because of apoptosis when they're maturing from being tadpoles. It happens a lot, and it happens in C. elegans. And thanks to the work of Dr. Brenner and a hell of a lot of abused grad students, the position of every one of those cells and the lineage of every one of those cells has been mapped. We know exactly where every cell in an adult C. elegans comes from, which means you can start looking at the genetic effects on development. Say we change one gene, what does that do to the timing? What does that do to cell numbers? And we know the genome, the complete genome, was sequenced back in 1998. The genomes of some relatives have been sequenced. And this is what it looks like. Up there at the top, you see ABPRAA. That's a code for a particular cell. And right at 100 hours, those are the numbers that are listed to the left. That's number of, is it hours? It's got to be hours. I don't think it's minutes. Okay, it's time anyway. Right at about 100 hours, cell ABPRAA divides into cell ABALPPP and ABPLAAP, and so on. Every time you see one of those branches, it looks like a cladogram, but it's not. Every one of those branches is a cell division event. We know when they happen. We know that some of the cells go on to apoptosis. You can see those branches that end in X's represent cells that die. And then you can see when the cells finally mature. Most of these go on to form a group of cells called the ring gang. I believe that's cells in the nervous system, in that nerve ring. And so we know cell by cell exactly what C. elegans is doing. And we can look at what the effect is of genes on the process and answer developmental genetics questions at a very nuts and bolts level, really answer questions as to how is it that genes guide the process of a single fertilized egg transforming into a complex adult. And this is what embryos look like. You can see some early cell divisions at the top. And then all the way to the adult. There's a hermaphrodite in which every cell nucleus has been fluorescently stained. So yeah, we can use C. elegans, a free-living nematode, to ask and answer some very basic questions as to how genes work together to generate, an adult body, to generate morphology, and how mutations in those genes might mess with shape and function. So even if you never mess with the parasites again, nematodes have been incredibly important in modern biology. C. elegans is a free-living nematode. I said I wouldn't say much about plant parasitic nematodes, but here's one. That's a soybean field under attack by Heterodera glycinase. You can see, the yellow patches of dying soybean plants are infected with plant parasites. They'll parasitize just about everything. There are some that will go after your tomatoes. And if you've ever heard that it's good to plant marigolds with your tomatoes, the reason is that marigolds supposedly secrete an anti-nematode chemical, which will keep them away from your tomatoes. I have not put that to the test, but I have no reason to doubt it. I had a student once that worked part-time as a groundskeeper for one of the golf courses in Conway, and mentioned that nematodes were one of the things that he really, really hated, because nematodes will use their stylets and pierce plant root cells and slurp out the contents and kill the plant. And you can have these patches of dying grass showing up right in the middle of the fairway of the 13th hole. And we cannot have that. I knew someone once who worked on a turf farm over in Perry County, a place that grows those square mats of grass that you lay down when you're starting a lawn. They had problems with nematodes there. Any crop can be attacked by nematodes. The animal parasites are no fun either. Sorry, I should have warned you. That's a piece of human intestine. This patient has just been operated on for infection with the human intestinal roundworm Ascaris. Not only does that cause, malnutrition and other symptoms, but if you have a bad enough infection, they can block the intestine completely, and that's an immediate emergency. So yeah, that's a rather graphic example of an animal parasitic nematode. But some of these nematodes are actually quite beneficial. On the left, that used to be a fire ant, Solenopsis invicta, Latin for the thing with a wedge-shaped face that cannot be conquered. Invicta means unconquered, but this one has been, because what has burst out of its abdomen is a number of parasitic nematodes in a family called the Myrmithidae. And these have been studied and have been actually have been artificially raised as a means of controlling harmful insects. There are myrmithids, for example, that will go after mosquito larvae. And at least for a time, there were experiments with raising myrmithids to attack mosquito larvae and cut down mosquito numbers. As you can see, there's been some research done into trying to control fire ants using myrmithid nematodes. And you can actually buy parasitic nematodes to control garden and agricultural insect pests. This is a picture I swiped off of Amazon. You could order these today if you really felt like it. There were a blend of 3 different species: Heterorhabditis bacteriophora, Steinernema carpocapsae, and Steinernema feltii. Each one is more effective against a different group of parasites. And off the bat, I think Heterorhabditis is good against grubs. Steinernema feltii might be particularly good against fleas. I can't remember the exactly what they're good at, but the mix is good against a very wide number of pests. And this is used by organic gardeners. And it's used in what's called IPM, aka integrated pest management. Integrated pest management says, don't try to treat your agricultural pests by bombing them with more and more and more pesticide, because they'll develop resistance, because that natural selection thing gets you all the time. And it can have bad effects as well, dumping too much pesticide in. You might use certain pesticides, but you should also use a combination of strategies, like growing resistant varieties, rotating your crops so that you don't get a chance of a parasite population building up in the soil because you're growing the same thing year after year, and use various, anti-parasite parasites to kill off your kill off your pests. So that's called integrated pest management, and these beneficial nematodes are useful in that sort of strategy. Okay, that's the general stuff about nematodes. On Friday, I will get to talk about Viking poop. I know you will not want to miss that, and I will stop recording.